targets, yeah. Our plan is to search for targets. We are planning to see targets, yeah. But we have to take into account what we name biodistribution, and so that is very special, yeah, that is very important. And when we are trying to figure out is that normal or it has a lesion or disease, every time we look at these pictures, we need to have this idea of norm. And it's special. And if you are not a specialist in radiology, especially in R&D, it's kind of tricky to do it. So pay attention to this. And we'll have only kind of acquainted with that idea, and we'll look in more detail at the picture, which is kind of really popular. And this is this image, yeah, this is what the name 18-fluorodeoxyglucose, or for short FDG. I highly recommend you to memorize, yeah, this name of the radiopharmaceutical, because this is the most popular and the most widespread radiopharmaceutical
Glucose, fluorodeoxide.
in our practice. And even I'll give you some hint to recognize this procedure and these pictures when you'll see them, and that can be really helpful because, yeah, if we think that they are around 90-95% of all modern R&D procedures, this is kind of helpful. So this is 18-fluorodeoxyglucose, but this is glucose. The basic thing, this is glucose. That means this is sugar. Yeah. So what does it mean? That means that we can see all tissues with like sugar, like very high uptake degree, and mostly who's the most sugar lover in our brain. So if you see brain like this, or like this in cover skin, that means this is FDG study. You will never make a mistake because this is the only one image in R&D which has brain like this, yeah, like in a really high uptake color, yeah. So keep this in mind. But not only brain likes sugar, yeah. What likes sugar also? Liver, spleen, heart, salivary glands. So they are what the name parenchymatosic tissue. Well, if we look at the lungs, yeah, they are partly tissues, partly air. And so they like sugar, but the uptake is less, yeah. So, and what is more, yeah, we can see partly intestines, yeah, because, yeah, we drink this sugar or eat. I don't know how to pronounce it, better drink or eat, and so that's why we see this. Or stomach, for example, we can see, and the sugar is excreted through urinary tract, yeah, and they see urinary tract and urinary bladder, all at the same picture, and that means background. So we have to keep this in mind, yeah, because this is known, yeah, and it's a little bit different from person to person because people have brain with different activity, different function of liver, and different activity and different perfusion of myocardium. So this is a little bit different from person to person. So this is important in analyzing this picture. And so if we try to analyze, for example, this example picture, yeah, so this is again this study, yeah, and this is fused PET-CT image, yeah, so the background, yeah, the black and white stuff is CT, yeah, and this is color uploading all these PET information, and the color scheme is this metallic one, yeah, feel this, yeah, so, and this is metallic one, then you see high uptake in this color, this is metallic scheme for color coding, yeah, and see what's there, yeah, so this is liver, yeah, this is liver, this is spleen, this is stomach, and this is like background, yeah. So this is air in a stomach, and it doesn't have sugar, yeah, and it doesn't eat sugar. These are lungs. Well, they like sugar, but not in smaller degree. So they are like black, yeah. So no color along background. And what's this in the liver? 2 lesions, and this lesion is hematoma, and it doesn't eat sugar. It could be seized with fluid, will be the same, or any fluid collection, any air collection. Then it's kind of like a little bit like a contrast enhancement. Yeah? And this is how metastasis looks like. Yeah. So compare, yeah, the liver. So liver, it's sugar, but in small degree. But usually, yeah, like tumors, they are really good sugar eaters, yeah, because they have to survive, they have to grow, yeah, and that's why they are eating it so greedily, so avidly, yeah. So this is how we should think, yeah. And strange probably idea, but pay attention that this is about FDG. This is a radiopharmaceutical, the really wide metabolic affinity. But look at another picture, and there is a picture of very, very interesting, very, very special radiopharmaceuticals. Probably you remember we discussed it a little bit on the last class. These two prostate-specific membrane antigen, yeah. So this is highly specific, yeah, really, yeah, like high targeted, yeah, radiopharmaceutical, yeah. But this is kind of protein which is in our blood, and it's interacting with all the body. And you see what is special. This is its normal distribution. Yeah. Does it look like background? It looks special, yeah, and it's actually compare. It's different background. Yeah? It's really different. Yeah? And so every time you have a new radiopharmaceutical, you have to keep that in mind, yeah, because you're comparing. Again, the same stuff. You can think about lesion only, yeah, if you know that this is not normal. And let's pay attention. What is normal, yeah, for this radiopharmaceutical? It's normal to be in liver, in spleen, in intestines. It's normal to be in kidneys. And it's normal to be in salivary glands, and even in lacrimal glands, what is very strange, yeah. All these tissues, they are excreting, yeah, these agents, yeah, and so that's why we see them. And people are different, and in some people, liver is less active, and this person has more active liver, and it's again different. So, yeah, when we are looking for diseases, we have to keep this in mind. And so, in this picture, only these dots, these are metastases, yeah, and only this is bladder, and this extra stuff, this is prostate cancer, and it's grown into retroperitoneal space, yeah. And so analyzing it, they have to know this. So this is very, very highly specialized. I found this picture like several days ago, and this was from an article, like pay attention, from Kenya. You know Kenya? Kenya is somewhere in the middle of Africa. And the trick is that it's Kenya, and that is PET study with monoclonal antibodies, yeah, for tumor receptors, yeah, and using this specific, very rare radioactive agent like gallium-68, and this is a very, very rare generator produce radioactive thing. And this is an article about specifics of prostate cancer metastases in Kenya people comparing with European people. I was kindly impressed. If you go to our R&D lab in our hospital, you will not find PET or even SPECT. In R&D we have two different approaches to both. They are about function and physiology, to study function and physiology. And they sound like dynamic and static, and usually when they see that something is dynamic, we think about function. But then they see that something is static, we don't think about function. But in this situation, this is not the right way to do, because they are both about functions, but functions can be different. Yeah? So one functions are quick and they are connected with changing, yeah? And other are slow and they are mostly connected with eating, uptake. I will not decipher this is not important for you. Just pay attention that this is something really simple, like what. Right now it's just in more big detail. So again, yeah, we have liver, and when we have intrahepatic biliary tract, and when we have gallbladder, and when we give some stuff and the gallbladder is contracting, and we see that how the stuff is going into the intestines. Well, this images are a little bit later, and so right now we can see there how it's going on even through the intestine, yeah. So that's how it's going on. So it's a dynamic study because it's a process, yeah, and we can follow it from the beginning to the end, even commanded it in some degree, just compress, yeah, these are 2 or 3 pharmaceuticals, one like the most used FDG thing, and a little bit for a couple for comparison. So if I speak about FDG, so FDG, it's kind of really popular and widespread, since it's really is useful in several different important situations. Yeah. First of all, yeah, we used FDG in tumor studies, in tumor staging, first of all, and it's really interesting idea why it's so useful and why do they use it so often. Yeah, the idea is that this glucose, like deoxyglucose, it can be utilized using classical aerobic way, in tissues with good blood supply, good oxygen supply, and the same thing, could be utilized. This form of glucose could be utilized by cells which are with low oxygen supply. For example, for tumor cells, basically, when the tumor is growing, what's going on? The cells are getting bigger in number and the vessels are behind. And usually when tumor is growing, it is like mostly, these cells are in situation of kind of ischemia. And that means that they are utilizing glucose in anaerobic way, And so this FDG thing can be useful to visualize both, And so that's why, we like to use it in looking for, let's name it, tumor burden, So how many tumor cells are in our body? So this is very helpful. Well, I don't say that we can find 100% tumor cells. This is impossible. And some tumors, they are not so avid to glucose, they're less avid. And sometimes inside the tumor we can have regions with different affinity to glucose. But basically, on big numbers it works, And if you look for clinical guidance for several important tumors,